Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everyone, welcome if you're new, welcome back if you're not. Today's episode is about glucose metabolism. We're going to focus on glycolysis in the TCA cycle this time, and next episode we'll discuss alternate things that can happen to glucose depending on the energy state and the energy needs of the body. So we kind of have to take a half a step back before we even start, because the first step in metabolizing glucose is obtaining it. We get our glucose from carbohydrates, mostly complex sugars called starches that we eat. And starches need digestion before they can be absorbed. And our friend who's going to help us out with that is amylase, starting with the amylase in the saliva and helped along by pancreatic amylase in the intestines. Amylase basically chews up the polysaccharides, which are long chains of sugars, by breaking alpha-glycosidic linkages. The beta linkages in cellulose can't be broken down by amylase, which is why we can't digest cellulose, which is a different story altogether, but just thought I'd throw it in here. And so anyway, you end up with an oligosaccharide, which needs to be broken down even further into simple sugars by the surface enzymes which are resident in the intestinal brush border. Now, there are specific enzymes that deal with each specific sugar. There's no way you need to know this at this point, but I'm just putting it here so that when we repeat it later when we talk about stuff in the GI unit, it'll be familiar to you. So lactase breaks down lactose into glucose and galactose. Maltase and isomaltase can both break down various sugars into glucose, and sucrase breaks down sucrose into glucose and fructose. So you end up with basically glucose, fructose, and galactose to be absorbed by the intestinal brush border. Now, uptake from the GI tract lumen into the intestinal cells and then out the other side into the blood both happen via transporters. So we're going to mention the two types of glucose transporters that we have. These two main classes of glucose transporters are the GLUTs, and these work by facilitated diffusion, so they work only with the concentration gradient, and the SGLTs, which are coupled symporters with sodium. So these are found in both the intestines and the kidneys, actually, to help extract all the glucose from the meals we eat and to be able to excrete glucose in the urine. So like we said, absorption in the intestine is going to use the SGLT transporter where 2 sodium and 1 glucose get pumped in, and the sodium gradient is maintained because sodium is actively being pumped out by the sodium-potassium pump, which uses ATP. So this glucose acquisition is an energy-requiring process, but indirectly. The glucose moves from the intestinal cells out into the bloodstream via GLUT transporters, and then from the bloodstream into the cytosol of the target cells, also via GLUT transporters, and these transporters use facilitated diffusion, so they only work with the concentration gradient once again. For the most part, GLUT transporters are going to be moving glucose into a cell, but in the absorptive epithelium, concentrations are so high inside the cell that it actually moves them the other direction into the blood. But regardless of where it's moving them, really the only thing you have to keep in mind is the concentration gradient, because GLUT is a very simple transporter. It just works the way you need it to based on concentration. And there are about 14 family members of the GLUT family. But we're only going to talk about 5 of them when we're really going to only talk about 2 of them. But let's talk about GLUTs 1 through 5, which are the best characterized GLUTs. So GLUT 1 is found in pretty much all cells. It's basically just the housekeeping transporter. GLUT 2 is liver and gut specific. It has a very high Km, so it's almost never saturated. So no matter how much glucose you throw at it, it's going to keep working. GLUT 3 is found mostly in neurons, and it's a very efficient transporter. That's kind of its hallmark. GLUT 4 is found in fat and muscle cells, and it is insulin dependent. This is important, and we will come back to it, so I'm not going to dwell on it right now, but just keep it in the back of your mind that GLUT 4 is unique because it's insulin dependent. And then GLUT 5 is found in the small intestines and the liver, and it's actually not a glucose transporter at all. It's a fructose transporter, but because we had already named it GLUT 5 by the time we figured out what it did, the name just kind of stuck. Things work like that sometimes in biology. It's weird. I don't know. Now, one thing I just want to mention, go back to GLUT 2 again for a second. The reason that the high Km of GLUT 2 is really important is because it's not saturated at normal blood glucose. So it can keep taking up glucose into the liver cells, let's say, even as blood glucose levels rise. So now what do we do with glucose once it gets into the cell through a GLUT transporter? The main thing we kind of think about is glycolysis. And the idea of glycolysis is to turn glucose into pyruvate, which can then go into the TCA cycle, but it intersects with many other pathways, which means that the carbons that are moving through the cycle can end up in many different places. So ATP generation is the largest thing we think about with carbohydrate metabolism, but we can also store the carbohydrates by turning them into glycogen. Another fate is to turn them into fatty acids and deliver them to adipose tissue. All cells can also turn glucose into fatty acids for their membranes, or carbohydrates for their membranes, various metabolites along the pathway can also be shuttled into different pathways to form amino acids, glucuronidates, they can enter the pentose phosphate pathway, etc. So the reason why I'm telling you all of this is that for you to realize when you regulate a step of glycolysis, you're going to be determining how all of these other reactions get affected, because depending on whether glycolysis is moving forward or not, different substrates will be in different concentrations, and that will affect how they move through other pathways as well. And these pathways are differentially regulated in different organs by the overall hormonal control of the body. So let's just remind ourselves before we jump in what the various tissues are doing in a fed state. So in a fed state, insulin is high, glucagon is low, because remember, glucagon gets activated when the glucose is gone, when the body is fasted. So in this situation of high insulin, well-fed, liver is going to be using glucose to generate ATP and also using glucose to store as glycogen and exporting it as fat. The adipose tissue is going to be taking up glucose, turning it into fatty acids, and muscle is going to be taking up glucose to fill its glycogen stores, which it will use if blood glucose is low. Now, in the fasted state, the liver is first going to break down its glycogen to send out glucose to the blood, and then once the glycogen is gone, it's going to do gluconeogenesis. Its big role is to maintain blood glucose level. That is like the point of the liver in the fasted state. In the fasted state, adipose tissue is going to stop taking up glucose so that it can go preferentially to the brain, and muscle is also going to reduce its glucose uptake, and it's going to rely on either fat or its own glycogen stores to generate ATP. And again, I'm just going to emphasize this because it's important. What's unique about the liver is that it can package and export fat to other cells, which other fat-creating cells don't do, and also that it can maintain blood glucose and do gluconeogenesis. The liver is the main blood glucose supplying organ when we're not getting glucose from our food. So let's talk about the big picture overall reaction of glycolysis. Basically what happens, 1 glucose gives you 2 pyruvate, 2 water, and 2 net ATP. It really produces 4 ATP, but it uses 2, so net 2 ATP. And it has 2 phases. In the first phase, you put in 2 ATPs, so that's the energy input phase. And then in the second phase, you're going to generate 4 ATP and 2 NADH, that's the output phase. These 4 ATPs that are generated by substrate level phosphorylation. So the ATP production is independent of the mitochondria and can be done under anaerobic conditions. And then the pyruvate at the end gets shuttled into the TCA cycle, which is going to generate ATP by oxidative phosphorylation. Now, sometimes the pyruvate can go to other places depending on the energy state of the cell. But what we're talking about here is how we generally think about the glycolysis pathway flowing forward, which is that glycolysis creates pyruvate and pyruvate goes into the TCA cycle and generates lots of ATP by oxidative phosphorylation. So now to go back to where we were, glucose-rich blood is hitting the liver. Now what? So GLUT2 is working to take up all this glucose, and as the glucose enters the hepatocytes, it's going to get phosphorylated for 2 reasons. Number 1, once it gets phosphorylated, it can't leave because phosphorylated glucose is not a substrate for the transporter, so this traps the glucose inside the cell. And number 2, this also maintains the gradient. Because the phosphorylated glucose is not a substrate, it doesn't count for the gradient. So the glucose comes in, it gets phosphorylated, and then all of a sudden the concentration gradient keeps pulling more and more glucose in from the blood, even though there's already a lot of glucose inside the hepatocyte. And this is how the liver acts to quickly buffer blood glucose so that we don't get these big excursions in blood glucose hitting our organs, because our organs really do not like that. So let's talk about this enzyme that does the phosphorylation. This enzyme that phosphorylates is called a hexokinase, and we actually have 4 different hexokinases. Again, hexokinase 1 is a housekeeping hexokinase. Hexokinase 2 we see more in muscle and fat. Hexokinase 3 we actually don't know much about. And then hexokinase 4, which is also called glucokinase, is the one that's going to be important in the liver cells. And it's mostly expressed in beta cells of the pancreas and in the liver cells. So how are these hexokinases regulated? Hexokinase 1, 2, and 3 regulation is mostly by feedback inhibition. If you don't need the glucose for anything, you don't really need to take up any more glucose. And if it's not getting used for anything, it stops getting phosphorylated so it can go right back out. Hexokinase 4 is different because it is not feedback inhibited. So in the pancreas, hexokinase 4 is important because it's actually the sensor that beta cells use to monitor blood glucose level and determine how much insulin is released. But we'll get back to that when we talk about the pancreas in 2 episodes from now. But in the liver, which is where we're focusing right now, like we said, hexokinase allows for this blood glucose buffering because as levels rise, we can take in lots of glucose before it hits the main venous circulation. Since you have GLUT2, you can take a lot in very quickly, and because hexokinase 4 has a high Km, it gets phosphorylated and it keeps it inside, and it's not feedback inhibited. And this buffering occurs independent of insulin secretion because insulin takes a second to start being secreted. This buffering happens without insulin. So now we're going to talk about the specific regulation of glucokinase because we said it's not feedback inhibited. Instead, it has this very cool and interesting regulation system. So normally the glucokinase is held prisoner by a glucokinase binding protein, which binds to the glucokinase and sequesters it in the nucleus. And this process is going to be regulated by the amount of free glucose, because the more free glucose that's allowed into the cell, the glucose is actually going to block the binding proteins from binding to glucokinase, and then more of the glucokinase is going to be free. When the levels of glucose are high, and they're blocking a lot of the binding protein interactions, they're going to be drawing more complexes out of the nucleus by diffusion, because the number of complexes in the cytoplasm is going to be low because the glucose is not allowing the binding protein to complex with the glucokinase. And this makes more hexokinase available to phosphorylate the glucose. I may say hexokinase and glucokinase interchangeably. Again, glucokinase is hexokinase 4, so technically it's probably more correct to either say hexokinase 4 or say glucokinase. But if I say hexokinase in this context, know that I mean glucokinase. Okay, so then as levels of free glucose drop, more glucokinase gets sequestered because the binding proteins are not being blocked by the glucose, and this slows the phosphorylation. So therefore, in a fasting state, when blood glucose is low and the liver is going to want to dephosphorylate and release glucose back into the bloodstream, you're going to have a condition where you've mostly put away the phosphorylating enzyme in the nucleus because the free glucose levels are low, and this is going to stop a treadmilling inhibition, and it's going to allow you to release free glucose into the blood when you need to. There is some downstream feedback regulation, so when intermediates of glycolysis like fructose 6-phosphate collect, they can promote the hexokinase sequestering by increasing the binding. And conversely, fructose 1-phosphate, which is an intermediate of fructose metabolism, actually promotes binding protein dissociation, so you get more hexokinase. Turns out that if you have a glucose meal with a little bit of fructose, it'll actually metabolize the glucose faster because it encourages uptake of glucose. Who knew? Alright, now we have to talk about glycolysis itself, and I promised we wouldn't get super granular, and we're not going to, but we do have to talk about some key intermediates and regulatory steps. So let's walk through super quick, and I bet it'll be better than you remembered, because I'm not asking you to track carbons or remember any structures. We're just going to talk through it, okay? So we can break it down into phase 1, which is the energy input phase, and then phase 2, which is the energy payoff phase. And there are really only a couple of steps that we're going to have to pay a lot of attention to, and the rest of them we're going to mostly ignore. So starting with phase 1, energy input, step 1 we already talked about. That's the phosphorylation to trap the glucose in the cell. And that's where you spend your first ATP, because remember, phosphorylation takes an ATP. Step 2 is just an isomerization, which changes glucose 6-phosphate into fructose 6-phosphate. Step 3 is one of the ones we need to pay attention to. It uses an enzyme called PFK1, phosphofructokinase 1, and you know what this is going to do because it's a phosphofructokinase. Yep, it puts another phosphate group on using another ATP. So now we have fructose 1,6-bisphosphate. Step 4, we split this 6-carbon molecule into two 3-carbon molecules, DHAP, dihydroxyacetone phosphate, and G3P, glyceraldehyde 3-phosphate. Step 5 is kind of cheating because it's just transforming the DHAP into G3P, so now we're going to have two of the same, 2 glyceraldehyde 3-phosphates. And this is the end of the energy input phase. Now we're going to enter the energy payoff phase, in which we're going to generate 4 ATPs. So step 6, a phosphate group gets added, but not by a kinase, so we don't consume any ATP, and we actually produce 2 NADH and 2 molecules of 1,3-bisphosphoglycerate in this step. Step 7 produces 2 molecules of ATP by taking back a phosphate group and putting it onto ATP, and we're left with 2 molecules of 3-phosphoglycerate. Remember, it was 1,3, and we took the 1 off of 1, so now it's just 3-phosphoglycerate. Step 8 moves the phosphate group over, so we end up with 2 molecules of 2-phosphoglycerate. Step 9 removes the water and gives you phosphoenol pyruvate, also known as PEP, and this is going to be important a little bit later. And then step 10 uses another important enzyme, pyruvate kinase, to make pyruvate, and this step also gives up 2 ATP. There it is. That's all of glycolysis. I'm sure it took you like many lectures in undergrad to go through it, but I just went through it in like 1 minute, so you're welcome. And when we talk about the important steps, we'll re-highlight them. But let's begin at the beginning. PFK1, phosphofructokinase 1. Why is it so important? So once the glucose is phosphorylated in that very first step, it's trapped inside the liver cell, but it still has a lot of options open to it. There are a lot of different pathways it can go through. But PFK, which phosphorylates it again and makes it a double phosphorylated fructose, commits it to the glycolysis pathway. That is the importance of PFK1, is that it commits the G6P, which is interconverted fairly freely to F6P, fructose 6-phosphate, to the glycolysis pathway. So how is it regulated? Because when things are important, they're always regulated. So there is cell intrinsic regulation, which is that high levels of AMP, adenosine monophosphate, which is indicative of a low energy state, activate this enzyme because it's going to want to drive the glycolysis pathway forward and generate more ATP. Whereas ATP and citrate, which are signs of having high enough energy, are going to inhibit it. So you don't need to go the glycolysis route, and you might want to use your glucose for other things. As energy depletes, AMP is going to go up dramatically, and it's going to drive phosphofructokinase to drive the glycolysis forward. And this kind of regulation occurs across all cells. Now, there's liver special regulation of PFK, which is kind of another regulation on top of that. And that is, we have to bring in a cousin of PFK1, which is PFK2. Now, PFK2 interacts with fructose, and it has both a kinase and a phosphatase activity. I know this is confusing, I'm sorry, but it can produce either fructose 6-phosphate, which is the same as that substrate that gets converted by PFK1 and feeds into glycolysis, or it can produce fructose 2,6-bisphosphate, which activates PFK1, which governs this very important step of committing glucose to glycolysis in the liver. In a fasting condition, what do we think we're going to want to do? We want to stop glycolysis, right? Because we don't have enough glucose. We don't have enough glucose to be creating energy. We need glucose to go backwards and give us blood glucose. So in a fasting condition, glucagon is going to work through a GPCR to activate pKa, which we've learned about before, which then phosphorylates PFK2. And this drives its phosphatase function, so you create lots of fructose 6-phosphate and deplete fructose 2,6-bisphosphate. Now remember, fructose 2,6-bisphosphate was helping drive glycolysis forward. So if you deplete fructose 2,6-bisphosphate, as happens with glucagon, this inhibits the flow of glucose towards glycolysis by slowing down PFK1, which is important because if we want gluconeogenesis, we want to reduce forward flow so we're not committed to the glycolysis pathway because we want backward flow. In the fed state, the opposite is going to happen. Insulin signals are going to increase the kinase activity of PFK2, which will increase the levels of 2,6-bisphosphofructose, which will drive glycolysis forward. So it's important to recognize this PFK1 point is a control node for hormones to control the flux through the glycolysis pathway. So not only is there cell level regulation, but there's also hormonal whole body level regulation at this point. Now I want to talk about fructose for like a half a second because fructose metabolism does impact glucose metabolism. So GLUT5, we talked about briefly, is that transporter that's needed for fructose to get inside the cell. And the fructose also, kind of like glucose, needs to get phosphorylated so that it commits to metabolism. The enzyme that does this is called fructokinase or ketokinase. So you need both the GLUT5 transporter and you need that kinase in order to get fructose to be metabolized. And fructose gets metabolized to DHAP and G3P, and those sound familiar because they're glycolysis intermediates. So once they get to that point, they can actually just hop onto the glycolysis pathway. The reason why this can be useful to us is because it's regulated differently than glucose. It doesn't use PFK1, which is the choke point in glucose metabolism, so it can kind of bypass this regulation entirely. And additionally, like we mentioned before, fructose 1-phosphate, which is an intermediate in the metabolism, helps get more glucokinase into the cytosol by interfering with the hexokinase binding protein situation. So a cell that has glucose and fructose will metabolize the glucose better than if it just had the glucose alone. But you're actually going to get more fat produced. It's more glycogenic. All right, now we mentioned this before, but I want to just mention this again quickly, that there is organ differential regulation. In the skeletal muscle and in the heart muscle, the regulation of the forward pathways are different. So in the muscle, in the fasted state, muscle is going to break down its glycogen for energy, but you don't want to release it. So its PFK2 is not responsive to glucagon. So glucagon is not going to change the forward progression of glycolysis because muscle is not able to do gluconeogenesis and you don't want it to go backwards. You want it to be able to use its glycogen. So this whole glucagon insulin regulation of PFK2 is only applicable to the PFK2 in the liver cells. It does not apply to PFK2 in muscle cells. Because bottom line, the liver needs to make and release glucose when you're fasting. So you want to inhibit glycolysis and upregulate glycogenolysis and gluconeogenesis. But in muscle, you actually want to enhance glycolysis in the fasted state using its glycogen stores because it can't secrete glucose on any other tissues, but it has to be more conservative in its uptake of glucose, so you want it to use its glycogen stores. Now, another heavily regulated part of the glycolysis pathway we have to talk about is the very last step, pyruvate kinase. Pyruvate kinase is regulated at the cell level in two different ways. It has feed-forward activation and it has feedback inhibition. So fructose 1,6-bisphosphate, which is one of the early glycolysis intermediates, is going to activate pyruvate kinase, which comes in later in the process, and it's going to kind of prime it. So when you have fructose 1,6-phosphate, it's like yelling down like, hey, glycolysis is starting down here, get ready. and it primes the pyruvate kinase to be ready to act on the phosphoenolpyruvate and create pyruvate. On the other hand, we have feedback inhibition similar to the rest of glycolysis, where ATP is going to inhibit this enzyme because you have lots of energy. Maybe you don't need to make more. Maybe the glucose is better used in another way. So just to go back a little bit and talk about how the fate of glucose is decided. So the first thing is the hexokinase action. Either we keep it in the cell or not. If the hexokinase doesn't phosphorylate it, it goes back out to the blood and we're done. Once it's determined that it's going to stay in the cell and it's going to be metabolized in one way or another, PFK1 and pyruvate kinase determine its fate. So PFK1 is the one that commits it to glycolysis, and if not, it can go to other pathways. It can go to the pentose phosphate pathway, it can go to glycogen production, which we'll talk about. There are a bunch of other things that can happen to it if it does not get committed to glycolysis, so that's where PFK1 determines the fate, and then pyruvate kinase commits it to the OXPHOS pathway by transforming PEP into pyruvate in an irreversible reaction. Now, making it into pyruvate doesn't necessarily commit it to the OXPHOS pathway, because there are other things that can happen with pyruvate. So once we have pyruvate, what can we do with it? It can be converted to acetyl-CoA by pyruvate dehydrogenase, and it can go forward into the TCA cycle, which is kind of what we would expect it to do. Or it can go backward by pyruvate carboxylase. It can be converted to oxaloacetate, which is a TCA intermediate, and is also involved in gluconeogenesis if the need arises. Because acetyl-CoA cannot be taken directly back to glucose, as we'll discuss next time when we talk about gluconeogenesis. So if we do need to create more glucose, it's going to have this pyruvate carboxylase take it to oxaloacetate. Or we can convert pyruvate to lactate. Now, why in the world would we convert pyruvate to lactate? There's always going to be some flux of pyruvate through the lactate pathway, just more or less depending on oxygen levels. And the reason for this is actually very interesting. So it's going to go to lactate almost exclusively in the absence of oxygen. And this occurs fully in the cytosol. And the reason this happens is because of the ratio of the NAD plus to NADH in the cytosol. NAD cannot get across the mitochondrial membrane, so it can get depleted, and it needs to be regenerated by donating electrons to the electron transport chain after it's converted to NADH earlier on in the glycolysis cycle. Without oxygen, the electron transport chain is not running, and so you need another way to regenerate NAD plus in the cytosol, because otherwise NADH collects, and this stops glycolysis. If you remember, there was this step in there that generated NADH. So if you don't have any NAD plus, you can't generate NADH, and things are going to get backed up at that step. So luckily, this buildup drives the conversion of pyruvate to lactate, which is energetically favorable, and this regenerates NAD plus, and then glycolysis can start up again. Now, while this is energetically favorable, it's extremely inefficient because you're only getting 2 ATPs per molecule of glucose out of substrate level phosphorylation versus in the electron transport chain where you get 20 plus. All right, so let's go back and talk about what happens in maybe the ideal state where acetyl-CoA is generated. So acetyl-CoA actually is generated in the mitochondria by pyruvate dehydrogenase. What happens is it releases CO2, generates NADH, and remember that we had a 3-carbon chain before. So now we get rid of one, we now have a 2-carbon chain. And acetyl-CoA production is regulated by feedback inhibition. If you get a buildup of CoA, which this is going to be important when we talk about gluconeogenesis, which is why I want to mention it here. If you get a buildup of CoA, it's going to stop pyruvate from going forward into acetyl-CoA production, which makes sense. But there's also hormonal regulation. So there are kinases that can phosphorylate the enzyme complex and reduce its activity, or phosphatases which can enhance its activity based on hormonal signals. So glucagon is going to want to decrease glycolysis, and insulin is going to want to increase it. And I'm just going to make this point again. There's always going to be cellular level regulation and hormonal regulation at all of these important points, and usually they're pointing in the same direction, but there's always kind of this double layered regulation. Now, what can happen once you have acetyl-CoA? The main thing is it can go into the TCA cycle, but it can also be made into ketone bodies in a fasted state where we don't want it to go forward, but acetyl-CoA can't directly be converted back into glucose, and it's also energetically expensive to go kind of the roundabout way and make it back into glucose. We can make it into ketone bodies. And the brain can use these ketone bodies. It doesn't like to, it prefers glucose, but it can use them in a state of prolonged fasting. And acetyl-CoA can also be used anabolically to create fatty acids, and we'll cover that in great detail when we talk about lipid metabolism. So let's talk about the TCA cycle, and I'm really not going to do it any justice at all. I'm literally going to say nothing about it. If you're a kindred spirit to me and you love to draw the structures and track the carbons, don't let me stop you. Do it. Do it in colored markers. It's beautiful, it's relaxing, and it's fun. And I encourage you to totally do it if you enjoy it. But the bottom line that you need to know right now is that acetyl-CoA, which is a 2-carbon chain, enters. It combines with this oxaloacetate, which is a 4-carbon molecule. Along the way, we lose 2 carbons in the cycle as CO2, and then we're going to be back to a 4-carbon oxaloacetate at the end because it's a cycle. The real products of this are not oxaloacetate per se, because you really need oxaloacetate to start. Again, this is a cycle. So the real products are the electrons that go to the electron transport chain carried by NADH. We also do generate GTP by substrate level phosphorylation, and you get FADH2 as well. The energetics of this cycle are fairly balanced. It depends on the individual reaction. So the delta G is going to be positive at some points along the way, but you consume the products enough to drive the reaction forward. And this keeps the delta G not super negative either, so that it can go backwards if it needs to generate intermediates at some point. Citrate, for example, is an intermediate in the TCA cycle and is super important for a lot of things. So you want to be able to bleed it out of the cycle and use it to create different metabolites like fatty acids for cell membranes, for example. So because of this, the TCA cycle regulation is not that complex. The pyruvate to CoA transformation is heavily regulated, and then running the cycle depends on having CoA, but once you get into the cycle, it's pretty much immediate regulation. So high energy is going to inhibit it, it's going to slow it and allow intermediates to be bled off, because if you think about it, if the cell is in a state of high energy, we don't need to be frantically producing ATP. We have enough time to relax and bleed off some intermediates and be anabolic, build things up. Low energy is going to drive it forward because our biggest priority is keeping the cell going before we build any new structures. High calcium is also going to drive the cell forward because in muscle contraction, you're going to have high calcium to generate contraction, and that accelerates the cycle because you're going to need more energy when you're contracting the muscles. One just piece of terminology to know around this is an anaplerotic reaction, which is a reaction that's basically run to replenish intermediates. So amino acids can be interconverted to feed the cycle, and the opposite is also true. You can bleed intermediates off of the cycle to convert to amino acids and use in biosynthesis, like we discussed in a high energy state. Okay, so that was part 1 of carbohydrate metabolism. We talked about how glucose gets to the liver, some of the tricks the body uses to buffer blood glucose, and we pretty painlessly, I think, talked through glycolysis in the TCA cycle, some key regulatory points, both intrinsic cell level feedback and hormonal regulation, and we talked about how we determine the direction of flow of metabolites. Next time, we're going to talk about the other thing that glucose does in the fed state, glycogen synthesis in the pentose phosphate pathway. We'll also talk a little bit about gluconeogenesis, how we reverse the irreversible steps of glycolysis to generate free glucose. Thank you so much for listening. I hope you enjoyed, and if you did, please consider taking a moment to rate the show and mention it to your friends and fellow students. I'd really appreciate it. You can also feel free to reach out to me at any time with questions, comments, or requests. My email is medtogether26@gmail.com, and I would be so happy to hear from you. Catch you in the next episode.